Snyder, and I'm going to be talking about my project that is determining the proliferation and differentiation potential of clonal human articular cartilage progenitor cells. Despite the limited regeneration potential of articular cartilage following injury, articular cartilage progenitor, also known as ACP cells, have the ability to produce stable articular cartilage in vitro. These have been isolated and cloned from the superficial zone of the tissue in recent years. Our lab has also found that there is a subset of ACPs residing within the deeper regions of articular cartilage. The aim of my project in particular was to compare the expansion and differentiation potential of clones from the different zones of articular cartilage. In order to do so, individual cells were isolated and expanded from both the superficial and deep zones of normal human articular cartilage. At 20, 40, and 60 population doublings, clones were then stimulated to undergo chondrogenesis in 3D pellet culture for 14 days. We wanted to determine the matrix production of these pellets. In order to do so, we measured the glycosaminoglycan, also known as GAG production, hydroxyproline, which is a surrogate marker for the total collagen content, and collagen 10 production, which is a marker of hypertrophy or ossification of cartilage. At 20 population doublings, there was increased matrix production in the superficial compared to the deep clones, as noted by the increased glycosaminoglycan and increased hydroxyproline content in the superficial compared to deep clones. There was no differences between the superficial and deep pellets at both 40 and 60 population doublings. For collagen 10 production, there was an increase within the deep clones at 20 population doublings compared to superficial clones, but this difference was not observed at both 40 and 60 population doublings. We also wanted to determine the percentage survival of clones at 20, 40, and 60 population doublings for both superficial and deep zone ACPs compared to the original number of clones that we started out with. So for the 20 population clones, there was about a 75% survival at 20 population doublings, which fell off a little bit at 40 and 60 population doublings, but significantly less so than the deep zone clones. Less than 40% of deep zone clones made it to 20 population doublings, and only 11% of those made it to 40 and 60 population doublings. So, in conclusion, these data reveal the differences between populations of chondrogenic progenitors residing within the different zones of normal human articular cartilage. Superficial zone clones have increased survival and propensity to form stable articular cartilage in vitro compared with deep zone clones. Differences seen within the matrix production at 20 population doublings are less at 40 population doublings and higher. This is likely due to the fact that the survival of only higher matrix producing clones that are less likely to hypertrophy from the deep zone clones make it to both 40 and 60 population doublings. Thank you for listening to my presentation.